Hello, everyone. Hello, can I have your attention? Okay. Last bit, I promise. All right. So I would like to introduce our first keynote panel of the conference. We've made it to the end of the first day. So this session focuses on the ever-evolving tapestry of biological design, urban informatics, and experimental fabrication research. These groundbreaking trajectories have emerged, challenging our perceptions of the built environment. The first delves deep into the confluence of architecture and biology, presenting a radical reimagination of design principles. Here, traditional architectural paradigms are recalibrated through the lens of bioinformatics, synthetic biology, and material science. This line of inquiry introduces the concept of biodesign, where living organisms are not mere bystanders, but active participants and collaborators in the design process. Structures envisioned through this lens possess the ability to metabolically adapt, akin to organisms responding to environmental stimuli. The implications are profound, suggesting a future where the built environment exhibits homeostasis, dynamically reducing the embodied energy of materials and crafting structures capable of adapting to climatic shifts and aberrations. The second trajectory centers on the synthesis of urban informatics and responsive fabrication. Here, the city is not viewed merely as a physical entity, but a cyber-physical system, deeply embedded within networks of data and computation. Materials and fabrication practices come to the forefront of shaping cities in an age of scarcity. Drawing inspiration from computational design, embedded systems, and urban ecology, this research postulates cities that are both responsive and predictive and that utilize ecology. This research postulates cities that are both, they utilize contemporary waste streams. Through decentralized data networks, urban morphology becomes a dynamic entity, constantly evolving based on real-time data and feedback loops. This vision shifts fabrication paradigms away from static outcomes, embracing a fluidity that is data-driven, adaptive, and aligned with material availability and optimized site-specific morphologies. Both these research avenues challenge entrenched academic dichotomies and offer fresh perspectives that can redefine the very foundations of architecture and establish construction methods. As we delve deeper into these ideas today, let us be prepared to question, reimagine, and embrace the transformative potential they present. When we fuse these two trajectories, we unearth a vision of the future where buildings and cities operate in symbiosis. The structures living and breathing through principles of biodesigns communicate and align with an urban fabric that's digitally attuned to its inhabitants and the environment. It is a harmonious feedback loop where biology informs data and data guides biology. This convergence of ideas signals new modes of making, challenging us to reimagine architecture and cities not as distinct entities, but as interconnected ecosystems, pulsating with life and technology. And with this, I would like to introduce our two keynote speakers tonight, Martyn Dade-Robertson and Areti Markopoulou. Martyn Dade-Robertson is a professor of emerging technology at Newcastle University, where he specializes in design computation, with a special interest in emerging technologies, particular to synthetic biology. Martyn's core research is an emerging field of biodesign, specializing in synthetic biology as a field that implies to engineering, design thinking from fields such as electronic engineering, computing science, and biological systems. In the UK, synthetic biology has been heralded as one of the 8 great technologies, which includes, for example, robotics, advanced materials, and fabrication practices. Martyn has led multiple research groups, including EPSRC-funded projects, computational colloids, and thinking soils, both concerned with developing bacteria-based sensors capable of detecting mechanical changes in their environment and synthesizing strengthening materials. He's also the founder of the Hub for Biotechnology in the Built Environment, HBBE, a new research center integrating design, biology, and engineering to develop new living buildings. The hub consists of more than 30 researchers and hosts 4 new research facilities, including an experimental house called the Martyn Dade-Robertson has degrees in architectural design, architectural computation, and synthetic biology. He studied architectural design as an undergraduate at Newcastle University. And later at Cambridge University, he did an MPhil and a PhD funded by the Arts and Humanities Research Council. His focus throughout the early part of his career was information technology, specializing in information systems, drawing parallels between the topological organization of physical and digital information spaces. His PhD, which was supervised in the School of Architecture and the School of Computing at Cambridge, was nominated for the Royal Institute of British Architects Research Prize and was published as a Routledge book entitled The Architecture of Information. He is currently the editor of a Routledge book series on biodesign and has completed his first book of the series entitled Living Construction in 2020. And with that, I would like to welcome Martyn Dade-Robertson.
So thank you, Assia, and thank you for inviting me to this conference. This is my favorite conference of all. So the first time I presented here was in 2016, and I presented a paper. Sorry, is that not in a good? Ooh, I'm a little taller than I. Sorry. And I presented a paper then on thinking soils, a project I'm about to talk about then. I think it's fair to say I was in the miscellaneous bit of the conference. I was in the one that the panel where no one quite knows where to put everybody, so they put those in that bit. So to go from there in 8 years to be a keynote, that's quite something. So I'm very proud to be here today and to be standing here with Areti as well. And I'm honored to be presenting. So my presentation today. So let me give you a brief explanation of our lab. So you'll have seen this logo quite a bit today. So it's not just me here. There's a number of us from this new initiative. Assia talks about it. Assia was part of it, in fact, was the concept architect for the OHM, our experimental house. So we've been building this up over the last 4 or 5 years. We split into different research themes. I'm not going to give you a full presentation on it, but if you are interested in the center, do talk to me and indeed my team about it at the end. I'm going to be presenting on behalf of one of the research themes, which is the theme I co-lead with Mung, who you also saw present this morning, living construction. I'm going to talk about through a few of our projects there. It helps, though, that I've had basically my presentation given this morning. So we've had 3 presentations from our group. We have 1 still to come, and I encourage you to stick to the end of the conference where Monica will be doing it. So it gives me an opportunity to go a little bit beyond the specifics of our projects and do what I like to do and take a bit of a helicopter view of the things that we do. So I'll mention bits of these projects, but I don't intend to represent them today. What I do intend to do is to talk about designing with agential matter and to describe really what I think agential matter is and why it might be relevant to this community in all kinds of different ways and why it's relevant to the way that we work now. And to do that, I'm going to put this idea of agential matter into a little bit of a context. And this is a context that I think this audience more than any really understands and will know about, but it's worth going through the history a little bit anyway, albeit in a very, very, very summarized form. So I'm putting agential matter in a classification system with 2 other concepts, inert matter and active matter. So this is a history, as I say, that we know well, but it's worth restating. So we have often in architecture, particularly in some of the formal traditions in architecture in the West, treated matter as something which is inert and is really through a heat, beat, and treat method, we derive forms, and those forms are idealized. So we have a mathematical idealized idea of form, and we simply try and choose our materials to occupy and to create those forms. This is true of the modernist era of architecture. It's also true of the early days of computation, where computers were very good at deriving forms, not necessarily very good at thinking and understanding materials. So that idea has been challenged in the discourse, and this is a discourse that's been led by this community through the philosophy of new materialism. Again, I'm showing what I think will be a quote that many of you will recognize, but that idea that new materialism is a new and emerging idea of thought that goes beyond this idea of materials following form, but thinks about materials and matter as form generation. And this has led to huge developments in architectural computation and creative and innovations throughout our field. So as well as it was really strange because this is going at a different pace to my screen. So I'm going, so this idea of inert matter now gives away to an idea of active matter. This gives away to matter which is empowered by its own tendencies and capacities, where we look towards what material and matter will do for us. And of course, we can innovate very directly in thinking about matter as being active. So I'm showing here an example of Skylar Tibbetts' work on self-assembly using hygromorphic materials. The capacity of materials to change in response to water is the basis for assembly. So where then does agential matter sit within this tradition? Well, it's really interesting because listening to the presentations today, I've heard at least a half a dozen mentions of materials having agency or matter having agency. So this already exists in our discourse. I'm taking my cue from researchers from developmental biology. So this is a paper by a developmental biologist called Jamie Davis and one Michael Levin, based in the UK and the US. Both of them are developmental biologists, both work on trying to understand how biology constructs and makes form. And this is a very recent concept for them, this idea of agential matter. In fact, the paper that I'm citing here only came out a couple of months ago and is the first formal citation I can find of this idea of agential matter. And it says that it goes beyond an idea of inert matter or even active matter to thinking, if we think through cells, we're thinking about a matter which has its own problem-solving capability, its own requirements, its own goals and agendas, which are rather different from those of the designer. This is particularly true in a field like developmental biology for people who are looking, for example, at tissue engineering. To understand the nature and dynamics of this new type of matter is a really major challenge and is still something that is we're quite a long way from understanding and fully realizing. The definition that I most like about agential matter, though, comes from a podcast. So I don't know whether anybody listens to Lex Fridman, his 4 or 5 hour podcasts are very good to listen to on flights. He does these deep dive interviews with all kinds of different people, often scientists. And he did a podcast with Michael Levin, with a large proportion of it was given over to this idea of what agential matter is. And in order to try to conceptualize it, Lex said, okay, this idea of agential matter, he said, well, if I think about inert matter, it's a bit like making things out of Lego blocks. So Lego blocks we think of being inert. I can say that my children are playing with Lego that is exactly the Lego that was bought for me more than 40 years ago. So, and I know it's pretty inert. It's doing the same thing that it was then. We can assemble it. It has a particular logic of assembly. And then if we leave it there, it will stay in that form and it will do what it's told. So that's inert matter. So if inert matter is like making with Lego bricks, then agential matter would be like making with dogs. So there are lots of advantages of stacking dogs to stacking Lego bricks. You think about it, the dogs can self-assemble, they could jump down and they could get back up again. You might be able to assemble them in different places. There are, however, some challenges with stacking dogs. And it's those challenges that I want to deal with today in our thinking. So how do we begin to approach agential matter, and what applications did it have in architecture? And I have to say, I was anticipating having to persuade you of this, but we've already seen lots of examples today where architectural designers are working with living cells as a fabrication strategy. We've all seen projects now by mycelium. So, and someone playing the mycelium drinking game can take another drink now. This is mycelium. The image on the left is one that we all know very well. This is David Benjamin, the Living's Hi-Fi Pavilion in New York, made out of mycelial bricks that have been compressed and make a load-bearing structure. The image on the right is one of my personal heroes, the first architect biotech entrepreneur. I think I would be right in saying that's certainly the way she's described. Ginger Dossier. That brick is made out of a cement that's derived from an enzymatic process created by bacteria, and is becoming now a really successful company making building materials at scale with microbes. So these are real hero projects, the hero projects of mine, but we're also encouraged in our group to think critically about them as well. And a voice that I channel quite often in our group meetings is that of Louis Kahn and the brick dialogues. I don't know whether anyone's heard of that, but the idea, Louis Kahn asked his students if they were short of ideas to ask a brick what it wants to be, and the brick wants to be an arch. And you go through that process. We'd have a variation on that, which is to say, well, do we want living cells to be bricks? So do living cells want to be bricks, right? And the answer is no. So if we take the same biological systems that I've just described before, this is more like what they want to be. So the image on the left is from one of my PhD students, Dylan Oskan, who's also one of the organizers, is over there waving at us over there. We'll come back to her work in a moment, but this is a mushroom. So mycelium wants to be a mushroom. No surprises there. The image on the right actually is really interesting. So this is a collaborator of ours, Ilana Kolodkin-Gal, and this is a microbial biofilm. So this is a biofilm created by microbes that make calcium carbonate. And the structures that you see on the surface there, she's demonstrated are formed with calcium carbonate. So if you knock out the genes that are responsible for the construction of that material, the construction of the biofilm, those structures will go away. And we've actually collaborated on a paper where we've proposed that these calcium carbonate formations are actually microbial architecture. What they're doing is making structures that give them defense, give them structure, and allow them to organize their community. So those structures you see, if we took away the genes, indeed, Ilana did this work, that make the calcium carbonate, those structures go away. So this is genuinely a kind of microbial architecture. So can we then make use of the tendencies and capacities that we see in biology to drive technical innovation, to drive a new way of fabricating these new agential materials? So there might be a few ways that we go about it, and I've got three. I do everything in threes, so bear with me here. So the first one, and this was my starting point, was to say, well, maybe we can get synthetic biology to do it. So why would we think we can get synthetic biology? Why might that be the best way of working with agential materials? Well, we've all heard the idea that DNA is the code of life, and sometimes it's described, particularly in the popular press, as the blueprint. And now as an architect, if I can get access to the blueprint, make changes to that blueprint, you would assume that whatever is constructed is going to be different, that I have some control over the final design process. That might be the best language for biology. Now, I'm going to give you a very brief, just in case there are people here who don't remember their high school biology, I'm going to give you a very brief explanation of this. So what does DNA actually do? Well, DNA is through a process known as the central, unfortunately known as the central dogma, because it sounds so dogmatic, but takes DNA. So we go from DNA through a molecular machine known as RNA polymerase to create RNA molecules, which are signaling molecules, which are information molecules. I'm desperately not looking at my biological colleagues right now as I'm completely mashing up this explanation of their work. Through a ribosome to proteins, and proteins do everything in our cell. They're the cell structure, they do the cell metabolism, they bring all together all of essentially that's what a cell is, this interaction between these many thousands often of proteins. But it's important to know that the DNA doesn't just code for proteins, right? So that's the coding bit, but DNA is part of a much bigger infrastructure, and we call those in synthetic biology, we call them biological circuits. Biological circuits start with a coding sequence, or at least have a coding sequence as part of them, but they also have these other components, and you don't need to know what all the components are other than that first bit, called a promoter, and we use symbols to describe them. They're very particular. So the promoter on the left is, we can consider that like a switch. It's actually the regulatory region for the genes. So in your cells, your genes are not firing all of the time. You're not making everything that the cell can make. You're turning on and off those different coding sequences depending on what your cells need and depending on what they're required to make. And so we can use those like switches, and so we can see that they have an input, and then we can derive then an output by switching over those switches. And then by putting them together, what we can do is build much more complex cell circuits. And so in the synthetic biology literature, there are all kinds of computational circuits that we can build. So we can make AND gates and NOT gates. We can do all kinds of formal logic using these DNA methods, and we can do things like create cyclical processes. So this was the basis for the first project that we really embarked on in a serious way. In fact, it was 2 projects that Assia mentioned, computational colloids and thinking soils. And the concept was really simple, turned out to be quite complicated, but the concept was really simple. What if rather than digging a trench in when we're doing on a construction site, we could get the soil to respond to loading by synthesizing a model that remodeled the soil underneath the construction and strengthened it. We wanted to do that with bacteria. So we hypothesized that we'd be able to engineer a bacteria that could detect mechanical stresses in the soil and then synthesize, in our case, calcium carbonate crystals to help bind the soils together and remodel them. That was the theory anyway. So one method to do that might be to have 2 different bacteria cells living in the cell. One, which is able to detect mechanical changes. And then the other, which is our builder cell, can then be signaled to using a chemical signaling between bacteria, and bacteria can do this. They have these chemical signaling pathways that we can use that will then create a material that will build something at the end of it. And we kind of did that, kind of. So we do in our lab now have on the left, this is actually quite a kind of old image, but it gives you an idea, a mechanical sensing bacteria. So it can detect changes in its environment, physical changes in its environment. And we see the way that we do that is we use a signaling molecule called GFP. If you've ever seen science fiction where there's a lot of GMO, genetic modification going on, everything glows green. That's because we use a green fluorescent protein as a signal to tell us that our genes are working, doing what we do. The image on the top, the top left there, the slightly less green ones are not being loaded, the slightly more green ones are being. It's not a perfect switch, but it's doing something of the job. Then the image on the right is showing bacteria, in this case encased by calcium carbonate crystals, so they're producing and actually nucleating new calcium carbonate crystals. We know an awful lot about how to do that now, and so we can switch on and off that process. So we haven't integrated it, but it's one thing that we've been able to do. So that's where I started my career. But there are challenges to this. To get to that point took us 8 years and over £1 million. In fact, this would be a lot over $1 million. It would sound much more in dollars. So over £1 million to get to that point. So it's very clear that our agential material, using this as the language to communicate with our agential material, is not the panacea for what we want to do. Just to give you a sense of what an actual cell circuit looks like, this is actually a pathway, a metabolic pathway of an E. coli cell, very, very simple bacteria cell. It's only part of that pathway. It's not a direct mapping of what I was saying before, but each of these nodes could be the equivalent of one of those gene circuits, and each of those lines is connecting those circuits together. It's an incredibly complex system, and even in very, very well understood organisms, it's not necessarily that well understood. So we're finding, for example, new gene mechanisms in very well understood organisms all the time. So to get and unpack that complexity is well beyond our current capabilities. And I go back to my dogs example. So if we wanted to stack dogs, would we genetically engineer them to do that? So we might. In fact, we've been doing this with dogs for quite a long time, right? So we've been making genetic selection through what's the word for it? Through yeah, exactly. Thank you very much. Yes, through crossbreeding and so on. We get certain breeds, so we might want quite a compact kind of block-like dog. We could probably achieve that. But actually, you're not going to engage with the dog in that way. You're going to have a different sort of communication, I would guess. So this brings me to my second concept, the hardware. So this morning you were shown a presentation of our ELF system. We have our mini ELF and our maxi ELF. And this is essentially a fabrication system that works with cellulose, and in our case, microbial cellulose. Just to give you a slightly different example of this, on the left-hand side, we have an electron microscope image of wood. On the right-hand side, this is microbial cellulose. Molecularly, it's about the same stuff. There's a little bit more going on in wood. We've got lignin and hemicellulose and other things, but fundamentally it's the same substance. But of course, the microbial cellulose is much more disordered, and that disorder gives us some potential material capability. But really, we would like to be able to engineer structures out of it, actually be able to make materials that don't resemble wood, because wood already does that, but it's at least organized to give us new and different material properties. One of the differences between a microbe making cellulose and a plant making cellulose is just the number of genes, the amount of information in the cell. So even a relatively simple plant cell has about 26,000 known genes in it, and they go even higher than that. There are examples of 40, 50,000 genes in plants. Whereas on the right, the microbe that we're using here has about just about 3,500 genes. So there's clearly a lot of information embedded in a plant cell that simply doesn't exist within a microbial cell. So what we do is that we try and build systems where we offload some of that information into computation. And this is the mini ELF version of what we were showing before, slightly different view of it. Sunbin, our PhD student, standing very proudly in the background. This is his first demonstration of the system. It's doing something very simple. Actually, it's a very complex piece of engineering to do something very simple, but it tracks the height of a growing pellicle and then makes modifications to the material in response. So what he's showing here, I can't go through the whole setup here, but what he's showing here is a complex set of nutrients that we're giving the cell. We're trying to make the cells as happy as possible. We're monitoring all kinds of different conditions, and then we're able to make these modifications dependent upon the growth status of the pellicle. So what he's been doing also is to think about what a design framework for this way of working might look like. And so offloading some of that information into our digital systems means thinking about a complex network of interrelated parts. In this case, what you see here is that on the bottom left hand of the image, you're seeing the bacteria that she's producing a biological material, but it's not just producing the biological material. We're also adding in there modifiers, and some of those modifiers are coming from other bacteria. So we have other bacteria that are interacting with one another to modify things. We then have a transducing process, which is trying to take a biological signal. In this case, it's just the height of the pellicle, but it could be, for example, the chemicals. We could use chemical signaling within the chamber. We can look at different chemical profiles, like things like pH and so on of the media. We then transduce that into a digital signal, which then goes back into an inducer, which tells the bacteria what to do. So in that way, we're building up a dialogue with the bacteria to try to move it in ways that we want to add complexity into those materials. And we do so in a very basic way at the moment, but the fundamental framework stands for much more complicated systems as well. Okay, so that's all very well and good, but I know the question on all your minds, okay? So we might be able to stack dogs, but what about cats? I believe there's a cat-based Instagram channel related to ACADIA. Okay, so post this. Someone should post this straight away. So I have an answer for that as well. It's the concept we like to call persuadability. So persuadability is a real thing. And also comes from the same source as that agential materials thing that I started with. Persuadability says that any biological system is liable for persuasion, but you have to know at what level to persuade at. This is Michael Levin's work again. So this was a paper that predates the agential materials paper, but it's still only 2022. So this is still pretty new stuff. And what Michael suggested is that one of the problems in biology is that we're doing a lot of hardware modification all the time. So all of that synthetic biology stuff, we go always for the hardware when we don't always need to go for the hardware at all. But actually, biological systems have many of these different ways of interacting with them. We think about plant cultivation, for example, changing soil conditions, changing their environment in different ways, right the way up to, and he considers this a continuum, to persuasion. So if I persuade you today, I've made a biological change to you. One way or another, your brain is doing something different to the way it did when it came in. That persuasion, if I have persuaded, you might not be persuaded at all. You may go out completely unpersuaded. But if I persuaded you, I've done, weirdly, a piece of biodesign. I'm not going to try to genetically engineer you to think in the same way that I do. I'm going to try and persuade you. And our biological systems are liable to persuasion so long as we're persuading them in ways that also fulfill their goals and expectations as well. So we've been experimenting with this in different ways. And I just want to highlight a couple of projects that aren't at this conference right now, but that you can certainly find publications on. So this is from, I wanted to call you my PhD student, Thora. You're no longer you have your PhD at this point. Thora had her Viva a few weeks ago. Yeah, I think that. Her examiner said this was a major contribution to the world of biodesign. I would say it certainly is. It's using the same process as fundamentally the same process that the brick that you saw, so Ginger Dossier's brick, but this is actually allowing the microbes to kind of find the form themselves. So it's actually mixing microbes with sand, pumping the bacteria, pumping the nutrients and reactants into the flow of the bacteria and forming structures based on where those forms and where those forms follow, and utilizing that process as a way of generating solid materials, but materials that have distinctive forms that are not exactly biological, but are somewhat different. The other piece of work I wanted to highlight is one I've already hinted at, and this is Dylan over there, sitting over in the corner. So this is a work that Dylan's almost finished her PhD now. When I've finished reading her chapter on the plane, we'll get there. Dylan has asked mushrooms, the mycelium, what it wants to be, and found the answer was mushrooms, but has also found that mushrooms are persuadable. So if you grow mushrooms in different conditions, different levels of CO2, different levels of humidity, different types of airflow, and using gravity, you can create many, many different mushroom morphology. We call this exploring the morphological space of the mushroom. It's based on two principles. One is the plasticity of mushroom growth. Mushroom growth is not one thing. There is no blueprint in its DNA. The same organism with the same DNA will grow in completely different ways given different environments. But also Dylan's work has shown it's predictable. We can actually predict how those different morphological changes will affect the, we can predict, and indeed have predicted, how the different environmental conditions will lead to different mushrooms to form and shape. It's also based on an idea that she describes as probability space. So this is the idea that, well, they do within reason, and kinda. Tolerances mean something very different in biology to the way that we would like them in a more engineered concept. So some things you can predict very well, some things you can't. Some things are liable to being very well predicted, but some things are highly sensitive to other conditions and other changes, and understanding that is a really important part of the work. This doesn't lead to architectural applications, by the way, but does lead to a way of thinking about materials, which we think is quite interesting. And then finally, I just wanted to show here the work of Aileen, who you actually saw her, Eileen, sorry. Now I've been saying Eileen's name wrong now for the past 4 years. I've discovered today. I've been saying Aileen and it's Eileen. I'm sorry, Eileen. So you saw today Eileen's work. Now, if Eileen's work looks like tissue engineering, it kind of is, right? So tissue morphogenesis is not just about genes, it's about the physical interaction of matter, air, chemicals, and energy. So the fact that she has things that start to appear like vascular systems in these very simple systems, this is not coded into the bacteria genes that she's working with. Even things that start to form like beating hearts are because this kind of morphogenesis is coming from the physical interaction of the cells and their environments in these really complex and emergent behaviors that are just fascinating to study. And I think her work has as much applicability in the world of tissue engineering as it does in design as well. So those are the ways that we're trying to work with these emergent forms. So what's the conclusion then of designing with agential matter? So I'm always a bit reluctant to add another term to our lexicon, right? So as a group, I think we make up too many terms for things that already exist. And actually, some of what I've described, it really does already exist. And we can find evidence of this kind of way of thinking about materials in practices that are not high tech, that are not of the now, that are of heritage. And I think we could talk about that in the conversation afterwards. But I do think it's a really interesting idea. And the fact that it's come and it's starting to become a thought in mainstream science means that I think we can make a contribution to that world. And in the same way, I think that world can make a contribution to us. So I think it's worth including in our lexicon. Taking an agential approach could lead to new fabrication strategies, but also I think might lead to new philosophies of material. Thinking about materials as agents, thinking about ourselves as agents, leads to a different sort of dialogue that goes beyond the brick dialogue that we saw in my Louis Kahn example, to a different sort of dialogue, a much more complex dialogue with our material condition generally. And cells are a really good instructor of that philosophy, I would say. And finally, I haven't really shown you any pictures of buildings. And I'm always feel very self-conscious when I do these things. I was an architect. I used to do buildings, and now we look at stuff in microscopes. So I think we're a lot closer, and I firmly predict that if I get invited back in 8 years' time, or I can get a paper in 8 years' time, and I'll be in the miscellaneous bit of the conference, we'll have built something of scale using some of these techniques. And we're getting there now. We're getting to the point where we can, but it is complex. Biology is hugely complex and difficult to scale for lots and lots of different reasons. And we're only beginning to understand that complexity, and working in this way, we're getting to bigger, at least things that I'm not showing you in the microscope, but we're not there yet. This is a question I think that we have to ask ourselves as a community is at what point do we join technology race? What do we do? Can we work on technology readiness level 1 when we really want technology readiness level 2 or 3? So this is something to think about. So those are 3 thoughts that I want to leave you with. Oh, and yes, and the thought that we probably shouldn't be building buildings out of dogs. Just before I finish, I'm going to do a little bit of shameless self-promotion because I have 2 minutes, so I can do this, apparently. So first of all, I want to give some acknowledgements. So I've given a shout out to some of my colleagues here. The ideas that form this presentation are the result of lots and lots of conversations with lots of different people. These are just the tip of the iceberg of the number of people we have in our hub. I think, Assia, you said we had about 30. So we were supposed to have about 30. We now have about 50 people in the HBB overall. So we're growing, and there's a lot of people I wanted to do, including our funders. So it's amazing that we get government funding to do some of these crazy things, but we are able to, and I'm phenomenally grateful to them because they allow us to do things that we didn't dream about a few years ago. I also wanted to, there's one thing I wanted to do just in the back corner there. And Assia is not going to, I don't know whether you're going to promote your book, but Assia has just had a book released. So I have a book series called Biodesign. Mine was the first one. Assia's is this, I think you're the second one. You got there just a second. Richard Beckett, who will be here actually, but was doing a video presentation, has the third. If anyone is interested in this area, particularly those of you doing a PhD who want to write a book form for this kind of work, we have an opportunity to publish those books. And I'm really keen. We have a pipeline for publishing the books that's really good. So if anyone is interested in publishing, they should come to me. I'm also setting up a journal. So you can tell I get rejected a lot for publication. So I just have to create venues to publish. So we have a new journal with Cambridge University Press called Biotechnology Design. There's a number of editors here, including Assia and others. We're just setting up. We have 2 questions, one on how to grow a building, and the others actually is, and the nice end to this story is, I reached out to Jamie Davis and Michael Levin. We've written a call, essentially a call to arms for those who are interested in agential material to add into this community by making papers for this new journal that we've created. And so we're excited, and those questions are live now, and I'd be very grateful if people wanted to contribute. I also just wanted to say we're hiring. So we're always hiring. A project I can say little about other than if you want to make buildings that by growing deer antler, you should have a conversation with me because we have a PhD studentship on that topic. And we have a couple of new, including postdocs and PhD students that we're about, PhD studentships that we're about to launch. I'm actually moving institution at the moment, so I'm going to be part of Northumbria University. So reach out to me now and I can give you help with that. But we're expanding our lab over at Northumbria, so we're doing some new work there. So if anyone is interested in doing that, then you should reach out to me. And with that, I think, if my clicker will work, that was my last slide. Thank you.
Coming back down. Thank you so much, Martyn. That was incredibly insightful and enjoyable. We're really pleased to have you here tonight with us. And I would like to introduce our second keynote speaker of this evening, Areti Markopoulou, who is a Greek PhD architect, a researcher, and urban technologist working at the intersection between architecture and digital technologies. Her research and practice focuses on redefining the architecture of cities through an ecological and a technological spectrum, combining design with biotechnologies, new materials, digital fabrication, and big data. She currently holds the position of academic director at IAAC in Barcelona, and she leads the Advanced Architecture Group, a multidisciplinary research group exploring how design and science can positively impact and transform the present and future of our built environment. She's co-editor at Urban Next, a global network focused on rethinking architecture through the contemporary urban milieu, and has been consulting and developing urban and research projects on topics including urban regeneration through data science, guidelines for urban technologies in public spaces, circular design and construction, and multidisciplinary educational models and in the digital age. Markopoulou is editor and co-author of Learning Cities, published by Actor in 2022, Edible from Tab, Black Ecologies, also by Actor, published in 2019, and Disrupting Through Circular Design from IAAC in 2019. She's the chair of Responsive Cities International Symposium that she co-founded in Barcelona in 2016. And she has served as head curator of international exhibitions such as Edible at Tab 2022, Future Arena on Site Robotics for Building Barcelona, Print Matter, Hypercity at the Shenzhen Bi-City Biennale in 2015, BB Make for Beijing Design Week in 2014, and My Very Own City at World Bank BR Barcelona 2011. In 2022, she was appointed the head co-curator for the Italian Architecture Biennale under the team Edible, or the Architecture of Metabolism. And with that, I would like to welcome Areti to the stage. Thank you very much.
Thank you, Assia. It's always weird to hear your presentation. It's also great to be here after actually many years. Last time I was at ACADIA was 2018 in Mexico. I came to receive the Academic Award of Excellence, which was a great honor. And then things happened like COVID, a child. So here I am back and I'm really, really happy. Thank you for the invite. I also have to be honest that I doubt it. It's a difficult moment to be here, especially with everything that is happening in the world. Of course, what is happening in Gaza makes us all really, really uncomfortable, or in Ukraine and other parts of the world. But I really hope that the work that we're doing all together as a community maybe will not save lives, that they are at risk because of weapons or political decisions, but they will definitely save lives because we are going to design and build a better world, at least in terms of environmental sustainability and social one. So, sorry for the hard beginning, but I think it's something I really felt important to comment. So today I would like to speak about discovering new resources for circular construction, discovering new resources that they would allow us to create that world that is more sustainable. I will use the concept of urban mining as a medium to drive design, but also to drive building towards a paradigm of abundance versus the one of scarcity, as the very same title of ACADIA this year mentions. I think that we are all aware of the impact of the built environment. The buildings are responsible for half of the global primary material consumption, while they are also accountable for almost 40% of global climate emissions. Now, 50% of these emissions are actually produced during or before construction. So although our practices, our policies, they are very much focused on creating buildings that they have less operational carbon, meaning less energy consumption or less CO2 emissions while they operate, the biggest challenge is actually to reduce the carbon emitted before the building becomes operative and after the life cycle of the building is over. So what is called embodied carbon. Now, embodied carbon is a challenge, but there is a second one that I would like to talk about, and that is a challenge related with the fact that the material balance of the Earth is being challenged. Now, the year 2020 was marked as the year that the total weight of human-made materials globally surpassed all the weight of life in Earth. And I think this is something that needs to make us think. And it is estimated, of course, that in the years to come, the growth of mass added to the Anthroposphere will increase exponentially. That growth of mass includes all the materials that we use in construction, but also, of course, includes waste. And we know that our construction sector is one of the world's biggest waste generator, at least in EU, but similar numbers in the world. The construction and demolition waste accounts for approximately 30% of all waste of the planet. So on the one hand, we need to build more for responding to the current challenges of population growth. I mean, we have reached 8 billion people just some months ago. But on the other hand, we need to do so by first making sure that embodied carbon is minimized. And second, material extraction and waste production is limited. So how do we do that? It is exactly at this point that we need to rethink resources, and that thinking needs to enter not only our practice, but also our education, our design culture, and our policy making for the built environment. Now, a proposal related to our work developed at IAAC in Barcelona, and part of the work that I am doing personally, is to operate with an abundance mindset rather than a place of scarcity. This is a new paradigm, which is especially relevant in the practices related to the design and production of the built environment, since it expands the definition of resources, where resources are coming from, where raw or non-raw materials can be found, and where can they be mined. So if agents that we are involved in design, planning, and construction process, we could actually shift the attention to the Anthroposphere as a source of, rather than just a destination of processed goods, then we will probably have the possibility to break that linear process of extract, use, dispose. Urban mining is a concept that could help us to do so. It's a concept that is useful to explore the material stock in urban systems, and of course the use of anthropogenic resources in new production cycles. Now, cities hold tons of materials that they are valuable, from waste to agricultural or textile industrial waste that could become new construction material, to demolition, of course, and construction waste. All this could be seen as value that could be reused. Now, today, I would like to showcase some projects that they are actually operating in 3 different scales. In the material scale, selected projects will explore different kind of bio or upcycled waste for the creation of new engineered composites that they could be used in construction. In the building scale, I would like to showcase some projects that they are actually using robotic technologies in order to scan and audit materials that they could then be reused when the building will be demolished. And then in the urban scale, with a series of Google images, satellite data, and machine learning, I will show how we can use these technologies to index the existing material stock and create urban material libraries. So within the concept of urban mining, waste is our future material. That is no doubt. I think much of the work that has presented today and will be presented in the next 2 days is going to be showcasing that. But starting from this material scale, we can observe that different types of urban or agricultural waste have the potential to become a new class of sustainable building composites through different processes that enhance their properties to match them with the building industry needs. So this is exactly what we're doing at Digital Matter. It is a research and experimental studio at IAAC that I lead for many years now. In some of our projects, we're working on upcycling demolition waste. I guess we all agree that construction and demolition waste is almost 1/3 of the total waste that is generated globally. And especially in Spain, we also have a lot of waste related to brick and ceramics. It accounts for more than half of the construction and demolition waste. And that means that 10 million tons of brick waste per year, they end up to landfills. So the problem of bricks, if anybody has worked on upcycling demolition waste, is the fact that you can it's very difficult to separate it from mortar. And that means that you either upcycle entire pieces of brick walls, as the exceptional examples that the Landanger group has shown in Copenhagen, or you can only use aggregates because the bricks are breaking down. And that means that these materials are downcycled. So they can either end up into landfill, but they can also be downcycled. So how can we actually upcycle them and bring them back to the construction is the question that we're working on that project. So we are basically working with additives, and we are generating aggregates and powder that could be used in order to create a geopolymer that is 100% out of demolition waste, and a geopolymer that could even become a binder to the same material itself. So we can create tiles that they are binded with the same material. And using Guastavino's Mansory construction technique, we can actually create arches or vaults. But we can also use molding techniques in order to create free form and complex form components that could be easily assembled and disassembled. And of course, implement them in different applications, building schemes and structures. And we can even eventually, because it's powder and aggregates, we could eventually 3D print the material and create ad hoc solutions and applications for the built environment. Now, these systems not only contribute to recycling efforts, but also offer alternatives in building construction that reduce reliance on energy-intensive materials. So if we really aspire to have a circular design and economy, then it is very important as well to start understanding where this material comes from, no? And that means that there is a need to start mapping in our urban environments where are the different demolition sites or where demolition would happen in the next years, and of course where new construction could happen. And that would allow us to create a map that is not only about the material itself, but more than anything about a new urban infrastructure of resources that could be reused. We will see a bit of that at a later stage of this. Now, sticking on the material scale, there are other projects that we're working on, such as, for instance, using cellulose and paper for creating modular walls or modular columns, and 3D printed to become different applications. Of course, cellulose is a perfect material for indoor applications, but it could also be used for outdoor applications in different kind of climates. And what it is important to realize here, this material actually doesn't have any certificate to go into construction. And that is one of the limitations that we have in research and academy, that very often, at least the materials that we want to mine or we are mining and processing in order to create new construction matter, it requires complex levels of certification that sometimes it's not very easy, but it also requires changes into the building regulations for allowing these materials to be implemented. The same material of cellulose has been used from some of our students and researchers in order to propose and create schemes in blind facades of Barcelona that would become actually houses for bees and increase pollination and bees population in Barcelona. bees have been prohibited in Barcelona. I don't know if you knew that, but it's a very interesting thing that is happening in certain cities. And continuing with some other materials, this is another research project that is actually going to be presented on Friday by Nicole Kirova. That is one of our PhD students and my colleague in some of the work that we are developing. And it focuses on creating structural building composites of biochar. Biochar is the byproduct of the pyrolysis of organic and agricultural waste, and it is therefore sequestrated carbon. So building with biochar buildings, it means not just buildings that they are zero carbon, but actually negative carbon, because they are buildings that they have sequestrated carbon in their nanoscale. So in this project, we're working on developing columns, beams, or structural walls that actually are made out of CO2 instead of emitting CO2. And Nicole is actually exploring different kind of manufacturing methods, such as casting for 3D printing. Casting could give the possibility of adding reinforcements to the pieces, but it can also tackle the existing industrial methods. So it's one of the most preferred ones for us. But Nicole is also exploring 3D printing that could offer the possibilities of creating functionally graded elements based on structural performance. And 3D printing could also be suspended in concrete pool, while casting can eventually happen in 3D printed formwork. Now, using topological optimization and testing the mechanical properties of the different mixes, it allows us to actually select the most optimum material mix for minimizing embedded carbon in buildings. Now, the most interesting part of this work, at least for me, is the fact that biochar is actually an excellent soil fertilizer. So when the life cycle of these buildings are actually over, then we can separate biochar and we can send it back to soil to fertilize it. And in a way, we are able to create an extended cycle of matter and a unique connection in that cycle among buildings, waste, and food, no? And we can therefore speak about buildings, or better said, building bricks, that they come from waste, they are negative carbon, and once their lifespan is over, then they can feed our soil for increasing agricultural production. I've been doing some research also here for Denver. Organic waste can also be mined to actually produce biogas. I don't know if you're aware, the ones that are from here, but Denver seems to not do great in terms of waste stream diversions, which means recycling. It only diverts 15% of its waste, whereas the national standards are about 30%. But Denver, or better said, the Colorado, has interesting initiatives on actually capturing methane from landfills and use them in order to replace fossil fuels or use it as a renewable gas fuel in vehicles. It's important to understand that methane, which is the byproduct of the anaerobic digestion of organic waste, is actually much harder than the CO2 emissions. So it becomes as important as minimizing CO2 emissions to be able to minimize methane or capture it. Now, within digital matter, within the material scale, we have more projects that we're trying to mine from different waste streams, materials that we try to mine. Some of them are using the waste stream of the cork industry in Portugal and Spain. Some others, the agricultural wastes of barley straw, the leftovers from the timber industry. And in a way, we're trying together with our students to support them to create their startups that they would allow them to make an impact in the markets. Now, I want to show this project because, it's not urban mined at all, so don't take me wrong, but it has been a project that has been a milestone for us to start rethinking materials not just in a very experimental scale, but also in a scale that would be able to be implemented in urban infrastructure or building scale. So that is a project that we did in collaboration with Acciona. It's actually the first 3D printed pedestrian bridge. And we have been working a year on that project in collaboration with the Polytechnic School of Catalonia in order to develop a geopolymer that would be able to allow us to print a structural optimum bridge. But of course, that bridge was initially planned to be placed in the courtyard of Acciona, which is one of the biggest construction companies of Spain. And then a month before the launching of the project, the mayor of Alcobendas, which is a city next to Madrid, wanted to bring that project into a public park. And this is the moment that everything changed, because Acciona didn't want anymore to take any risk of any sustainable material whatsoever. So we went back to use a normal concrete and cement. But that was indeed a project that for us has been a milestone to start developing more sustainable materials for construction. That project has been prefabricated and assembled in only 2 days. And although its materiality is not as optimum, we have a solution to make it much more sustainable. Now, beyond recycling materials and beyond using waste as future matter, we must not forget that there is an abundance in some of natural materials as well. So something that we seem we have forgotten when we got stuck in the revolution of steel and concrete, but earth is one of them. And of course, that is not new. Earth constructions have been significant part of the architectural production for centuries. And the work that we are doing at the 3D printing architecture program back at IAAC in Barcelona is to revisit earth construction and technologically enhance them for building affordable and circular housing. Together with a group of researchers and students, we actually developed an earth-based material that it is enhanced with bioadditives and that requires no firing. And that means that it is biodegradable, it is locally found. And since it requires no firing, it also means that once you build a prototype or a housing and its lifespan is over, then you can reuse that material to construct another one. And initially we started printing the earth mixture with a robotic arm that we have in our installations back in Barcelona. But then, of course, this was limiting us in terms of scale. And if we want to speak about the architectural scale and construction, that robot is not enough. And that made us think of different ways of how we can achieve and create a bigger machine. This is actually a cable robot that comes from one of our collaborators and partners, Technalia. We have been working with Technalia in another project, but when we were in their headquarters, we saw that machine that they used in order to basically paint aircrafts. And then we thought, hey, what about hacking that machine? What about hacking a machine like that, which is very easy to assemble, to put in a box and to send away? It's a cable robot at the end of the day. And what about attaching a nozzle that would allow us to print the material that we had, and it would allow us to cover a big area and therefore start printing big. So we did that, and they agreed to let us the machine. And we first went into a construction fair where for 3 consecutive days, we were printing in real time an installation just to showcase to the industries and the people that they were around that this is possible. And this has certain structural possibilities, and it also has certain aesthetics within it. The last achievement of this research that has been running for more than 9 years now is the one that you are seeing in this video. It's actually the construction of a one-to-one small house that we built in our forest campus inside a natural park. What we did was not anymore to use the hacked cable robot, but we collaborated and we partnered with WASP, that they are creating big 3D printers, and they let us bring a machine on site that we have now. It's our machine, and we are able to actually test the materials that we can find on site, and we can actually start printing big prototypes. It's interesting that this project has received a lot of different awards, but more importantly, the new European Bauhaus Award, that for us is very, very important because it showcases that the European Commission, the European policies, they start to finally recognize that these kind of projects are not just experimental, but they can start affecting the construction industry, and therefore start changing different kind of regulations to allow these applications to be marketed, no? To be implemented. We also had worked in the past with the UN Habitat to do projects that involve communities, because that is an advanced technique, but it's also a technique that could be customized and could be used by locals. So it's also interesting how we put the technology in the service of different communities and people. And then we have also created different kind of images, because what we also want to explain is that these solutions are not just for developing countries, but also, why not, for our Western society. I think Gaudi would really love that aesthetics. Maybe he would like to work with us as well. So, so all great and all exciting when we work with natural or upcycled materials, but what about the existing building stock that we can find in our cities, no? An exciting aspect of the circular thinking is that we can consider these buildings as material banks, and that we can work on different technologies of mapping, tracing, or scanning these existing materials to evaluate which ones could be reused, creating therefore alternative material libraries for construction. So one way to digitize matter in these buildings is through scanning technologies. One of the research lines of the robotics and advanced construction program that we run at IAAC is to explore the possible use of aerial and ground robots, that they are coupled with computer vision, and they are able to scan, they're able to sort, and they're able to digitally archive existing material stock in buildings. So once processing these data, it is possible to create libraries of different building elements and materials, such as, for example, concrete blocks, wooden beams, or brick walls, that they can be then reused in another building in the future. And there are usually two types of data set, one related to data scanned in pre-demolition, and a second data set which is related to post-demolition. So the three important steps of this process includes building inspection, classification and localization of the material stock, and last but not least, also geometry reconstruction, so that these materials could become forms, and therefore the form follows the availability of the resources that we have. Now, that's an image that has been made by our students, but it's an image that I always use, and I like it very much because such concepts and processes can introduce, for example, new evaluation criteria that we can set up for buildings. Till now, for instance, we are evaluating buildings on energy performance, for instance, using LEED certificates. But could we do something similar with the material aspect? That would give buildings a unique material passport or a certificate that describes the material value they have or they will have after their life cycle is over. And in order to do that, of course, we would need some new urban, and that is circular hubs where separation, process, and sorting of these materials could happen. Actually, circular hubs is now a very key concept that the European Commission is working. We have received funding to start understanding how we can create protocols of where, how, and how many circular hubs a city could need to have in order to start implementing circular economy in the construction. Now, is it possible to extend the tracking and the scanning of our material stock at the urban scale? I mean, it is impossible to talk about urban mining or about circular architecture and construction without actually thinking about the city scale. And this is what we did for this project that is called Urban Mining Material Library. It has been developed with a group of experts and a group of researchers with the City and Technology program at IAAC. And the project proposes an end-to-end process of an integrated web-based application that encloses and delivers cross-reference data about the material stock of a city. Initially, we started with a case study of Barcelona, and we use actually unstructured data from the Open Street View to identify relevant patterns for making estimations of the quantity of the state and of the projected availability cycle of different materials in building facades. We started with glass, brick, concrete, timber, but this can be expanded with more materials. And by applying predictive modeling, the algorithm can actually identify, it can geolocate, and it can quantify facade materials with a present accuracy of 87%. 87% might seem a good number, but it's actually not if we want to work with these libraries and materials. So to increase accuracy, we need to do an overlapping of the image comprehension and the GAN training with existing cadastral data, such as, for example, the building age or the land use of the building, and then start to create typologies that would allow us in a very synthetic way to have conclusions of where can we predict these materials in the different facades of the building, but also how we can expand that prediction not only into the building skin, but also what's in the interior of the building. But again, to me, the value of this project is not so much into the accuracy, but actually in the use of open unstructured data as a basis, because that makes it a highly repeatable application. And it's also, in a way, attempting to propose a solution that can democratize data or can democratize access to data for local decision makers in countries where open public information is not available. So we did start with Barcelona, and we are able to zoom in and to see the different neighborhoods, as you can see in the video, or even a specific building, and we can overlap data of the prediction of materiality with data on the building age of the building or the land use, and that starts to create a library that would probably allow decision makers to take more informed decisions about how to implement policies for circular economy and circular construction in cities. As I said, Barcelona was the initial case study, but it became much more interesting, and we presented that in the UIA in Copenhagen in July, when we actually did a sprint to manage to create 3 more cities, different cities. Singapore, Helsinki, and New Delhi was added to Barcelona in an effort to try to see what would that mean for areas that have different availability in terms of open data, even in terms of open unstructured data. You know, Google Images or Google Street View in Barcelona is not the same as in New Delhi. I will finish with this project idea concept, and I do that because usually when we are talking about how we make our built environment more sustainable, then some people or most people would say and would agree that the most sustainable building practice is no building at all. No? And of course we agree, but that's not always possible. Now, what it is possible and what we can do is to expand the lifespan of our building as much as possible. And in this idea, in this project, in a work with the Center International School of Design, we worked with some researchers and students for retrofitting a high-rise building in Shenzhen that was to be demolished and be built out of scratch. So instead of demolishing it as planned and constructing from scratch, we have proposed to reuse the existing structure of the building, saving huge amounts of CO2, and then to create a detailed catalog of the materials that could be reused for the renovation. So a series of different modules for housing or common spaces have been created, reusing existing entire brick facades, precast concrete panels, or even window frames and other furniture, with the goal to minimize the amount of the materials that we can bring on site for the construction. This is how the final design idea looked like. It expanded square meter of the building for creating open spaces. It used design to boost passive cooling, but as well as it radically changed, let's say, the hard aesthetics with a much more domestic recycled atmosphere. So yeah, no building is also a way of building. Now I want to sum up. We've seen a series of different projects, from using natural materials for housing to auditing existing building stock that can be reused, or even turning urban waste streams into construction matter. And it is clear that our discipline has plenty of possibilities to innovate and to rethink its operation by rethinking resources and where the resources are coming from. We also, of course, need to educate differently how the future architects can operate, where can they find their material libraries. And of course, we definitely need to educate, and that's going to be a hard job to do, the decision makers or the policy makers. Now, I would like to close this talk highlighting that the urban mining is a concept that drives us to reimagine our cities as vast repositories of valuable resources, as vast mines. And this mindset challenges our perspective from one of scarcity to boundless abundance. And this paradigm shift not only redefines how we actually perceive waste, but it might eventually also transform the way that we approach current urban development. So I'm really looking forward to the discussion on all these topics. I also want to leave you with two more slides, one that is about a series of books that I co-authored and edited relevant to the topics, but also I want to end up with an invitation to Barcelona to the Responsive City Symposium that is going to take place on 27th and 28th of November. The topic of this year's symposium is collective intelligence design, and we actually want to promote and explore a design paradigm that brings together different kinds of intelligences, human, machine, interspecies intelligence, and you're all welcome to join us. Thank you very much.
Thank you very much, Areti. I'll welcome both of you on the stage, and we'll start our panel discussion. We'll invite questions from the audience. I know we're a little bit pressed for time, but we'll be fast. Thank you. Oh, beautiful, insightful presentation. The pleasure of listening to tonight. I think you both raised incredibly pertinent points, and I suppose I'd like to pick up where Areti, you started off in terms of the difficult context that we're currently facing, but also matters of equity and pedagogy. You've both entered various realms that perhaps stray from what we've traditionally defined as architecture and architecture pedagogy, especially at a foundational level. But we're also entering this new era where some of the emerging tools may perhaps liberate us as practitioners to actually venture more into these realms. So I suppose my first question is, how do you think this is going to affect the wider architectural framework of architectural pedagogy? But also in terms of equity, how accessible is this going to be internationally within various contexts?
Yeah, that's a hard one to answer. So, one thing I would say is that the Thinking Soils projects and the Hub for Biotechnology and the other stuff that you saw there started off as a teaching initiative. So it was a master's studio that allowed us to do the experimental work. And I think a lot of us do that. It's somewhat unusual in architectural education, unlike other academic fields, where you don't do your most experimental work in your teaching place. You leave that outside, and so a lot of the early concepts came from designing studio. So that's the first thing. And actually, one of the things I've really missed in the last couple of years, I haven't had as much time to teach as I would have liked. In terms of these enabling tools, there is one of the things that we found about biodesign is there's this kind of two sides to it. There's this kind of very high-tech version of it, which is in labs and requires incredibly expensive equipment that nobody can really afford. Not even we can really afford them. And then there is a whole community of people who are just doing this in their kitchens and their bathrooms. And we have dissertation students who make incredible mycelium things at home. And there's something quite liberating about that. And one of the things that we've been thinking about is liberating the biological techniques as well. And so there's a great open source community. There's a release of all of these hardware and software methods. And we see lots of innovation in that community, and the biodesign community in particular is set up in that way, actually, to try and democratize those things, I think. So we see a lot of promise there. I think there's still an inequity of knowledge to some extent. I think that the collaborations that we do, we find a lot of people coming to us because they don't necessarily understand some of the fundamentals, and they've not had the education on fundamental aspects of biology and so on that are required. So I think there's a need to do that. And I think we're thinking about programs that will do that. But yeah, that would be my.
Yeah, maybe from my side, I think that there are two keywords on that question. The first one is open source. Martyn mentioned that. There is an exceptional community out there that they're sharing their knowledge, and there are platforms that they also help them to share that knowledge. And one peer is learning from the other. And I think that we need to find ways to reinforce these communities and also to equip these communities with skills and funds, for them to further operate. And the second word, maybe I would say, that is distributed education. I think this is something very important. We took a lot of lessons during COVID. At IAAC, we have been running a distributed program since 2007. We did that, of course, in collaboration with the Center for Bits and Atoms at the MIT, but it's the Fab Academy program, which is a program that is distributed and is connecting different fab labs around the world. And based on that program, we're also now setting up other distributed design and distributed educational programs that they have different levels of accessibility, from free accessibility to, the possibility of getting into different nodes in different places in the world and share all the knowledge. And I think that this kind of new example of distributed education could also expand and reach more people and allow new pedagogical models that have in their core inclusivity and equity.
And in terms of collaboration, you've both had a very extensive list of collaborators, both human and non-human alike. How do you envision the future of the architect? Well, what is going to be our role, in the emerging contexts that we're seeing in terms of resources, in terms of ecology, political context, etc.? We've seen a lot of shifts in the last few decades.
You should start.
I will. First of all, I think that the idea of star architect dies. Bjarke, I believe, is going to be the last one. I really admire him. He's a good friend. But it's going to be the last one, I believe. That idea dies, and I think that's fantastic, because we really need to to start establishing more collaborative ways of working. It's not anymore, the architect is not going to be about a decision or a wish of a form and try to materialize that form. It's not anymore about imposing form into matter, no. It's we are going to move towards a discipline of architecture that includes a lot of different other expertises. We already start to see offices that they have biologists, computer scientists, economists, ecologists. So architect will become the mediator of these different disciplines. And this is why we also need to be able to know a bit of everything so that we are able not necessarily to code ourselves. I don't code, but I do understand what code can do and what the different kind of softwares can do. And I think that this is something very, very important for the architect, but also for the discipline of design in general.
Yeah, I couldn't have said it that any better, really. That would be my sense as well, that the losing the ego, it's when you're sitting here as a keynote and you're saying you're losing an ego. I think there's a lot of that. I would say from a research perspective, we've done very well in being seen as being a serious research, fundable research institution, in part by pretending not to be architects. Because something that's very strange to me is that, so I pretend to be a synthetic biologist or a microbiologist or biotechnologist or whatever it is. The reason we're successful in the projects that we do is because we're partly because we're governed by a kind of architectural way of thinking, which we don't well articulate and can be applied in lots of things that to do with the built environment, sometimes outside it as well. But we never really describe those as methodologically, such that the scientific research community will take that seriously and then invest in it in that way. So what we do is we pretend to do other things. But one of the most, I think the moment, when I started my synthetic biology course to try and make this shift, I was clearly very nervous for lots of reasons. I was well out of my depth, and I was really helped by the fact that I opened the first page of a cell biology textbook. The first reference there was to Buckminster Fuller and to the transegrity structure. And Bucky Fuller came up with the transegrity structure as a design before it was discovered to be the way in which cells structure and hold form. And for that said something about that way of thinking, which I think we need to actually celebrate. So in a research context, we think in architectural ways, and that gives us a kind of, and I still don't know how to articulate it. It's a spatial, structural, material knowledge. There's something about process in there as well, but we don't articulate it very well. And what we're trying to do in research is actually to make that thinking come alive, to do things that are not necessarily designing for the built environment, that might end up in all kinds of other things. And so I feel there's something of a turn there. I hope that I think there are a number of people who are demonstrating that in our community, actually. And so that's my hope anyway.
Thank you. And I would like to open the discussion to our audience at this point, because I realize we're a little bit behind schedule, and I'm sure they have a lot of pertinent questions as well. So we're just going to circulate the microphone. I think we have somebody over there.
Hi, thank you both. Really amazing presentations. My question is more for Martyn. There was a question earlier today that I'm sort of going to steal, which was about killing the living system, particularly mycelium. But there was also a kind of question about maintenance. And I'm wondering, when I saw your presentation, you were talking about persuasion. I kept thinking of bonsai trees and how they require a certain constant persuasion to be a certain way. But in a way, we could kind of liken that to maintenance, right? Is that something that's part of what you're thinking about with these living systems? And if so, in what way?
Yeah, interestingly, I'm sounding like I'm promoting my book again. I'm going to say, in my book, I use the bonsai tree as an example of this. Actually, no, I don't use the term persuasion at that point, but that's that kind of cultivation, very close cultivation. So yes, we do. So there's a field of engineered living materials, which I think that's a central question of that, which is to say that what we would really like is for our materials to retain life and to continue, because functionally they can do all kinds of interesting things by staying alive. They can self-heal, they can change, and they can adapt, and so on. I would say the other thing that I've learned in biology is that the boundary between living and dead is not so clear either. So a work that we did in our group, led by one of our postdoctoral researchers, looked at the self-healing of mycelium after it's been treated. So mycelial leather composite she produced, and it was able to put holes in it, and it was effectively dead to all intents and purposes. Put holes in it, then re-nutrified the holes, and the material re-healed. And it was because that particular type of mycelium sporulates. Those spores are seeds, so it retains that capacity for regeneration in life. So one of the areas that we're thinking of is that middle ground where we get these materials that we need to be dormant and static. So with a mycelium leather jacket, you don't want mushrooms growing out when it starts to rain, but you also need to be able to, you want to retain those lifelike qualities. And so there are these gradients in this, and it's true actually of many of the structures. You saw the living room, the biologically knitted structure. That was effectively dead, but it carried on living. There was mold and all kinds of things growing on it. It provided a new habitat for new things, and we kind of want to celebrate that as well. So there is that. It is part of our thinking that an old fabrication method might just be to kill the material, but what can we do that maintains that aliveness to actually to do something useful for us and maybe for the environment as well?
Probably like the idea of care rather than maintenance, especially when we are talking about living systems, because, you know, we have a forest in our forest camp. In order to maintain the health of the forest, we need to cut trees down. No, that's part of the forest management or the health of the forest. Of course, I don't know how exactly it is in your case, but I guess, you know, like removing moss and trying to maintain, quote to quote, something alive is more like taking care rather than maintaining.
Wonderful. Well, thank you very much to all of you, and thank you to our wonderful keynote speakers. And we hope you join us for the opening reception further down the street in about half an hour. Thank you very much.